Very good morning. Today I will show you just something else you can do in the course of your bioinformatics research, and you need to modify your protein for easy adaptation to your work. First, we're going to start from here. Let's check our present working directory, pwd. Okay, I want to change to another directory. I already copied that. And then let's find out the content of this directory, ls. Okay, and that's what I want to load. So I'll just say load, then enter the name of the protein there. Okay, you could just type in, or if you want to copy, you can copy. Just ensure that the space in between is just 1. So load. Now we have the protein loaded. I will now come to display sequence, and you can see that, whoa, this is a very elaborate protein, and of course it is the fatty acid synthase, a complex enzyme. But for docking, for example, your interest could be just the ARO domain, and you want to pick only that specific part because this whole bulky protein may interfere with the docking process. Yeah, for computational efficiency and to avoid issues like electrostatic effects, so it is better to work with only the domain. But we know that this enzyme, fatty acid synthase, requires NADP or NADPH in the reduced form as a cofactor. So we'll try to look through how many chains we have here. So this is chain A, and then we look through down. Okay, this is chain B. Okay, so if we come down here, we have NDP. So those are the cofactors. All right, so we'll try to identify them. If you look at, okay, I'll change background of this now to BG color, BG underscore color, white. Is it better? No. Let's leave it as it was. I'll just take it back to black, BLACK. Okay, I think it's better like this. So we will now try to get just this part of it. We have 2 NDP co-crystallized, but we have to now look at which of them is within the ARO domain. So we can do that by simply isolating amino acids around the NDP, since it's a cofactor and we want it as a part of the ARO domain that we want to use for docking. And this is important in case we need to compare it with other enzyme forms. Your work may not be directly like this, but you can still learn and use the same approach. So we will come here and create a section. Let's see, let's start from, say, 2000. Let's say this is 2021, 16, 11, 6, 2001. So let's start from here, and we just select all of these up to this point. Okay, we can see now that of the 2 NDPs, the cofactors, one has been selected. Okay, let's deselect again and then see exactly what transpired. Okay, I'll start again. This is the first one, and this is the second one here. So looking at it, we have from 2000 down. Okay, so it means we do not need this other one out here. So I'll take this one out. Now it's no longer selected. Remember, what we're trying to do is save a part of the entire protein that will be useful for our work. But then if we save this one out, Okay, let's start with this one first. So what we have here now is what is now here as select. For PyMOL to be able to do any work here, we have to first of all create this section as an object here before we can save it out. So we'll say create. Let's call this one. The enzyme is 8VLE. I'll just say 8VLE section. Okay, let me use underscore, please. Sec, comma, from selection. That's select. Enter. So automatically it has created the object here. You can see 8VLE sec. But we want to save only this part out as PDB. So what we will now do is to say save, let's say 8VLE, oh my bad, I beg your pardon, 8VLE, let's call it section so that we can differentiate it in our working directory. 8VLE section, let's just say clean, CL as cleaned, .pdb, and then comma, save it from where? 8VLE underscore SEC. So once you enter, so save already wrote 8VLE. So we can check our directory again, ls, just list. So you can see we started with 8VLE PDB. Now we have 8VLE sec clean.pdb. And if I go to that directory, you can see it's here. So this is the original enzyme, and this is what we now have. But let's view to see if this is interesting for us. I'll use DSV so that we don't ruin what we have going in the other interface. Oh, now we see a very tiny section, and then we have the NADP hanging out. So we can go back and just get a larger portion of this particular enzyme. So let's now start from, say, 1971, and we select up to this. We already know for sure that this other NADP is not of interest. Let's try to pull this one in. Then let's take this one out. Of course, that one is gone. You can see it again. I don't need this one. I want the one within the domain. So we will now, we've created a new selection now. We go through the same process. I'll just call up the previous thing. Create 8VLE sec, let's call this one sec2, section 2, from select. So we now have the object here, 8VLE sec2. Then the next thing we want to do now is save it also. So I will just also call up all the previous, okay, save 8VLE sec2 clean PDB from 8VLE sec2. Just understand, and that's the interesting about PyMOL, how to use all of these. So we have created another section now, which is PDB. So I can say list again, ls, to show me all the components of the directory. 8VLE, this is the one we just created, and this is the one we created earlier. So we can also once again go back to the working directory here, and just to make it easy for us, that's why I'm coming through all of these. View this one now, section 2. You can see it's a bigger complex, and we have something like this. So you just select what you want to ensure that you have a good representation that can be useful for your work. This is particularly important if you have to create a section from a multi-enzyme complex. I hope you find this video useful, and I wish you the best in your bioinformatics research. Thank you.